With the next speaker, Professor Galon, we are continuing this combat of cancer with the immune system. And you are from the famous INSERM organization, Institut in Paris. And I've read your CV, your short CV. It was all in French. But I could see. Yeah, I only know one French sentence. This takes time. But and once in a conference with Patrick Couvreur, sitting there, I see him there. I translated my name in French. My name is Gert Storm. I said Gira de la Tempête. So I could understand that you do wonderful work, and you have many papers. You well recognized. We know that, of course. And please take the floor.
Thank you. So here are my disclosures. So still today, clinicians worldwide are categorizing patients with cancer according to the tumor cells, according to how the cancer has progressed, how large the cancer, how the cancer has progressed into the primary organ. So that's the T stage, according to the invasion to the nodes, the N stage, or the distant metastases, the M stage, and according to multiple tumor cell-related parameters of aggressiveness, like the grade of differentiation of the tumor, like the mutation status of the tumor, or the chromosomal instability, etc. And based on these categories, the patients are classified either early stage or late stage. It gives the severity of the disease, and the patients are treated accordingly. But what we found more than 18 years ago now, and that we have demonstrated in multiple publications, is that all these things and the tumor cell fate is dependent upon our immune system, is dependent upon our pre-existing immunity. In fact, all these key parameters, the progression of cancer, the invasion of cancer, the fact that there will be a recurrence of cancer, are all dependent upon the pre-existing immunity of the patients, which is determining the fate of the patients, if the patients will survive or not. And the pre-existing immunity is also determining the likelihood of treatment, and in particular, response to immunotherapy. And this is a continuum of cancer immunosurveillance. We believe that there is no escape. There is a constant dialogue between tumor cells and immune cells all across, from precancer lesions up to the metastasis, and I will focus on the metastasis side. And I will first echo what Lex Egermont said earlier. This is in a different cancer type, rectal cancer. And just for you to know, the standard of care in rectal cancer is also a neoadjuvant treatment, but with no immunotherapy. This is a neoadjuvant radiochemotherapy that is followed by surgery. Some clinicians, and that's the standard of care. Alternative care would be that some clinicians would do this neoadjuvant treatment, radiochemotherapy, but then would watch and wait and not do the surgery, waiting for. An alternative could be to do precision medicine. And the question here could be, can the local immunity guide surgery? And this is what we have looked at in colorectal cancer. We published several papers, and what we were interested in was to evaluate the immune status, the immune patterns before the neoadjuvant radiochemotherapy treatment. And this is a test that we have developed, Immunoscore, that quantifies the densities of cytotoxic T cells within the tumors before the treatment, the neoadjuvant treatment. And this gives a standardized assay and classify patients according to a score. So it's a linear score, and we can stratify patients in two groups if we want to. According to the score, you see on the x-axis this linear score, and on the y-axis, the likelihood of having a complete response to the radiochemotherapy neoadjuvant. And you see that the likelihood of having a complete response increases when you have a better immune system, when you have a higher immunoscore. And for those patients who have a complete response, now we look on the right side
at the 5 years survival of the patients according to this immunoscore, low in red, intermediate in orange, and high immunoscore in blue, pre-treatment. And what you see in that first publication is that 100% of the patients, if they have a good pre-existing immunity, a good immunoscore to start with, and have a complete response, they would have 100% survival. And therefore, we collected patients from clinicians who did this watch and wait attitude that didn't do the surgery. And these are two independent cohorts. Actually, this is the TTR and DFS, but we would have the same thing with two independent cohorts, a trained cohort and a validation cohort. This is an international validation cohort with 249 patients who were under watch and wait, no surgery. And these patients, as you can see, can be stratified according to immunoscore, low, intermediate, or high. And the patients who have a high immunoscore to start with, even though they had no surgery, had an excellent outcome for 5 years with no recurrence, showing that for these patients, neoadjuvant radiochemotherapy is enough, surgery is not needed, and they don't even need an immunotherapy. They have a good enough immune system to start with that the neoadjuvant radiochemotherapy is enough, and therefore for low-risk patients, now we have ways with biomarkers like immunoscore here to stratify the patients and propose alternative strategies. So instead of the standard of care, neoadjuvant plus the surgery of the rectum, the rectal cancer, which gives, of course, with this rectal surgery, a very poor quality of life with urinary dysfunction, sexual dysfunction, fecal incontinence, many issues. Now we could do personalized immunoscore-based treatment. On the biopsy, we can do immune bioassays like immunoscore, predict the complete responders, and then not do the surgery. This is organ preservation, better quality of life for the patients. The next is on metastatic patients, and I will give you some examples and why we think that there is no escape. And first, we wanted to see whether the current theories of cancer evolution in human would be valid. And in this article, we looked in space and time and how the tumor cells were disseminated from the primary tumors to the metastasis and then from metastasis to metastasis. And we took patients having multiple synchronous and metachronous metastases. And all the current theories of cancer evolution are all tumor cell centric. Basically, there are 4 models: the linear model, the neutral model, the Big Bang model, and the most accepted, the branch model, in which different driver mutations in different tumor cells would lead to a dissemination. But what is common is that all these theories are tumor cell centric, and none of them involve the possible role of the immune system. And so in human, we demonstrated that in fact there are essential parameters within each of the metastases that explains the likelihood of dissemination of tumor clones. And of course, we did all the exome sequencing to follow the evolvogram of the tumor clones and could show that there are different patterns of metastasis. These different patterns according to the immunoscore, the adaptive immunity, and according to the immunoediting of the tumor cells that are facing this immune reaction, and with several different escape mechanisms. One of which being the immune escape associated with the overexpression of PD-L1. And so we proposed an immune selection model as in human that could be demonstrated with this interaction between tumor cells and immune cells, a dynamic interaction which is ongoing in each of the metastases. And so we proposed that based on this pre-existing immunity, we could define alterations, immune alterations, and propose appropriate treatments to the patients in a more precision-type medicine according to the immune system of the cancer patients rather than only to tumor cell parameters. And this is what we did in this colorectal cancer patient study, in which we were mostly interested, of course, by the MSS, which represent 96% of the populations in MSS colorectal cancer. So this is a randomized clinical trial in first-line metastatic colorectal cancer patients that were treated with chemotherapy plus bevacizumab, which is an anti-VEGF, or the treatment arm, same thing, FOLFOXIRI, chemotherapy plus bevacizumab anti-VEGF plus atezo, which is the anti-PD-L1 antibody, the checkpoint. And in that study, what we did was to evaluate another Immunoscore assay, which is this Immunoscore IC, Immunoscore immune checkpoint, which quantifies not only the cytotoxic CD8 T cells, but also the expression level of PD-L1 and the distance between the CD8 and the PD-L1, because this is the receptor that is inhibiting the T cells. So it's a spatial information, distance information, clustering and proximity of these cells. And according to this score, Immunoscore IC score, we can stratify patients into two categories, high or low patients. And what you can see from these curves is that, in fact, the MSS, this is only the MSS population of patients. With the dotted lines, these are the Immunoscore IC low patients. And you see no benefit in the treatment arm between the two dotted curves. In contrast, the plain curves, you see in yellow the control arm, and in blue the treatment combo immunotherapy arm. And in that case, you can see that 32% of the patients with colon cancer MSS, in fact, are benefiting from this combo immunotherapy, and they are only the ones who have the appropriate pre-existing immunity as measured by this Immunoscore IC test. There was an update at ASCO showing that this is valid not only for PFS recurrence, but also for overall survival with a hazard ratio of 0.4. And this shows that 32% of the MSS patients with colorectal cancer would benefit very significantly from this particular combination of immunotherapy with very prolonged survival. The last example I'd like to give is for another type of immunotherapy treatment, that is CAR T cells. So in that case, you take the blood of the patients, you modify the T cells so that they would express a chimeric molecule, in that case CD19 CAR T cells, in order to target large B-cell lymphoma patients. This is the ZUMA-1 clinical trial that is the trial that led to the approval of the CAR T cells in refractory large B-cell lymphoma in third-line treatment. And in that study, we analyzed the pre-existing immunity, again, of the patients before the infusion of the CAR T cells. And there are multiple ways to improve CAR T cells. Of course, you can improve the construct, you can improve the intracellular signaling of the CAR T cells, you can find better targets of complex constructs. But one thing that you should not ignore is the fact that all the patients are different, and all the patients have already a pre-existing immunity ongoing. And this is what is illustrated here. These are the patients who underwent CAR T cell treatment. And these are two evaluations of their pre-existing immunity before the infusion of the CAR T cells. The Immunoscore, that is again a densities of cytotoxic T cells within the biopsies, and this immunosign, that is an immune gene expression signature. And you see in blue the high patients having a high pre-existing immunity to start with, high adaptive T cell, Th1 type cell, pre-existing immunity, showing that these patients do benefit much better for receiving the CAR T cells than the red patients who don't have this pre-existing immunity to start with. So on top of the T cell fitness that is used to make the construct, on top of the tumor burden, and on top of the construct that can be different, the pre-treatment and the pre-existing immunity as measured by biomarkers, measuring things about our immune system, is telling us a lot about the likelihood of response to treatment. And if you want to learn more, this is our view of cancer. This is an immune view of cancer in which all the blue bubbles are the good T cells on all these concentric circles, which represent all the many different cancer types here. And you see the good immune cells and the bad immune cells that are very valid in multiple different types of indication, showing that a cancer in the lung or in the bladder or in the colon is very similar according to the pre-existing immunity of the patients. And with that, I'd like to thank all the people in the lab and our collaborators, and thank you for your attention.